research, and I'm here today to introduce you to a new feature on our website called At the Bench, where we're producing short video segments to introduce you to novel technologies and methods that we hope you'll find to be interesting and useful in your research. Our first segment today is on multiplex cytokine analysis, and so it's appropriate that I'm joined today by our product manager for multiplexing, Dr. Chung. Good morning, Tom.
Good morning, Alex.
Tom, can you tell us very simply just exactly what is multiplex cytokine analysis?
Cytokine multiplexing is simply the simultaneously measurement of multiple cytokines in a single sample. Cytokine quantification is something that has been performed commonly in the field such as immunology or cancer, but it was traditionally carried out one cytokine at a time using a technology such as ELISA. In modern biology, we are often very interested in the modulation of multiple cytokines under certain conditions. So by using multiplexing technology, one can easily to measure several of your interest cytokine to as many as several hundred different cytokines in a single sample.
Tom, can you tell us what are some of the advantages of multiplexing over traditional singleplex analysis?
Sure, Alex. Multiplexing can offer huge cost saving over running multiple singleplex assays. Moreover, the labor saving can be very enormous. For example, using multiplexing technology, it can be fairly routine to measure 40 different cytokines simultaneously. But you can imagine that it would be a huge undertaking to do this analysis using traditional ELISAs. Additionally, if the sample is very limited, multiplexing makes it possible for you to extract massively information out of a single sample than with the traditional singleplex methods.
That's great, Tom. Can you tell us a little bit about how multiplexing is actually performed?
So there are two main technologies used in multiplexing, but both are essentially based on ELISA. For example, in the first type, which is the bead-based, performed in a 96-well plate like this one. So antibodies specific for individual cytokine are coupled to the beads of different color, and these beads are then incubated with the sample of your interest to allow cytokine to bind. In the following step, the fluorescently labeled detection antibody are added to the beads. These beads are then passed through a detection instrument. So by measuring the fluorescence intensity of these detection antibodies and by referencing it to each bead color, one is able to easily determine the cytokine concentration in your sample. In the second type, which we called is a slide-based, microscopic droplet of antibodies specific for the cytokine of your interest are spotted onto the slide, creating the array. The array is incubated with the sample of your interest. Then you watch the slide, and then the array is, you will add the detection antibody onto the array. After that, the array is imaged using a microarray scanner. So the fluorescence intensity at each location on the array is proportional to a specific concentration of the cytokine of your interest.
Are there any specific types of equipment that researchers need to have in order to perform either of these types of multiplexing?
Yes, that is something that the researcher they need to consider. The bead-based technology is primarily performed using a Luminex detection instrument platform. As a result, it's very convenient because the data acquisition and analysis are fully automated. However, if you consider to setting this up, you need to make sure that you have access to the instrument or you could consider purchasing one. On the other hand, for the slide-based technology, you need to make sure that you can have access to a microarray scanner. So for most of the time, if you have a genomic core facility in your institute, you probably can find a standard microarray scanner for use for this technology.
Tom, what advice do you have for researchers on choosing between these two alternate methods for their own particular project?
Like many things, one has to make a call on a case-by-case basis. But there are some very useful guidelines to help you with that. For example, if you have identified a small set of cytokines of your interest that you want to analyze, and let's say these analyzed are common molecules such as interleukins or interferons, you probably will find the Luminex bead-based technology to be the easiest for you. On the other hand, let's say if you do not have a strong idea of which cytokines that are important in your experiment, and then you want to cast a wide net, then the slide-based, the array-based technology could be superior, because the possibility to measure up to 1,000 different cytokines simultaneously.
And lastly, Tom, how can researchers find out more about multiplexing if they're interested in potentially applying it to their own research?
We at Jomar and CrossBio Lab are very glad to offer free consultation about which method of multiplexing may suit a given project, as well as to offer experimental design assistance. Researchers are welcome to contact me personally at tom.chung@jlresearch.com.au or call our office at 03-9764-2566.
Great. Thank you, Tom. Thanks for your time, and thank you for tuning in to At the Bench with Jomar Life Research. We hope you'll join us again next time.